Good evening, all of you. Myself, Dama Venugopal Rao. So I'm working as lead analytical R&D for Dr. Reddy's Laboratories. It's my pleasure to be here to present my experience on few of the critical excipients characterization in complex dosage forms. So it is essential to understand the Q1, Q2 of the excipients in complex dosage forms. The Q1 is the generic product shall have the same excipients as that of RLD, including the grades for some of the critical excipients. Q2 is essentially the concentration of excipients used in the generic product, which shall be within plus or minus 5% of the concentrations present in RLD. Excipients quality and concentrations may significantly impact the finished product CQS. For example, the difference in buffers and compositions may lead to change in osmolality and pH. Difference in polymeric excipients may lead to change in viscosity, drug release, which can eventually impact the product performance. Hence, any major changes in Q1, Q2 need to understand and thoroughly justify the impact on its product safety, quality, and efficacy. So these are the few examples of complex dosage forms, mainly the ophthalmic dosage forms, and where each of the product contains at least one polymeric excipient. In some of the products there would be more than one polymeric excipient as well. It poses very much challenges in quantification as well as the grades identification uh, as part of Q1 Q2 reverse engineering aspects. So it's basically many of the polymeric excipients. It undergoes degradation with time. So example HEC, HPMC, and few of the even smaller molecular excipients as well, like benzyl alcohol, sodium bisulfite, and also some of these critical excipients having a tendency to adsorb on the primary packing material. Example polysorbate, and also it's important aspect to understand its molecular weight, etc. So during the formulation process of milling micronization, it undergoes degradation or changes in the polymer properties. Hence, it is very challenging to understand it. And some of the critical excipients, like some of the critical products having carbopol, etc., are having solubility limitations, which again pose enough challenges for characterization and also the quantification. And another important aspect is when we quantify as part of Q2, uh, many times the reference product having a tendency for the with water permeation into the container, which leads to variations in Q2. Hence, it is important to understand these changes with time and to get the accurate information of Q2. And another important and very challenging activities. Polymers comes with a different molecular weight and different degrees of substitution, and understanding the right type and right grade is really a challenge with the suitable analytical technique. And another major challenge to the industry is lack of advanced analytical techniques such as size exclusion chromatography coupled with the multi-angle laser light scattering detector, or the A4F connected with the MALS to get thorough characterization of the polymeric excipients. And overall, it is very important to understand the chemistry of the molecule to understand the behavior of the excipient in the product, and also for uh, thorough characterization of the excipient. As an example, as a case study, which I took it as a hypermellose, which is a HPMC. This is the molecular structure of HPMC, where it is widely used in oral, ophthalmic, and topical formulations. And being this is a non-chromophoric, selection of chromatography is very important to understand the right quantitation tool, and also the grade, which is essentially so the molecular weight and substitution, which is very much essential to understand. And if if the industry, if the some of the companies doesn't have the right the detector to for quantification, usage of same grades of standards, uh, which is very much similar to that of the polymer present in the RLD, is essential to understand the right quantitation, and also to understand the molecular weight distribution profile, uh, by conventional GPC, it is very much essential to have structurally similar molecular weight standards, which gives the similar hydrodynamic radius properties to get this molecular weight information. As an example, which we have shown here, this part of the hypermellose, which is methoxy content and propoxy content, having same molecular weight grades or having different types when the substitution changes. So it poses enough challenges to understand these structural aspects of it. So this is one of the characterization technique which we have used for quantification of hypermellose in ophthalmic suspension. So where uh, the HPLC coupled with ELS detector is used as a quantification tool, and the peak against peak is quantified. Essentially, we know the importance, the requirements of ELSD, the mobile phases, etc. Should mobile phases and diluent should be volatile, and we use the Jorbach's SB 300 angstrom column, which gives um, enough distribution of the molecular weight in the column to have a Gaussian peak. And these are the experimental conditions which were used for quantification. Once we understand the right quantification, then important aspect is we have to use the right grade of the standard for the determination of the, basically to get the right response of the standard for accurate quantification. So to get the right the grade of standard, to understand the molecular weight. Here we have used SCC coupled with multi-angle laser light scattering detector to understand the molecular weight distribution profile of hypermellose, and where ultra hydrogel column was used with 300 mm 7.8 mm ID column, and with eluent as 50 millimolar sodium chloride to minimize the secondary interactions in the column. So the challenge here is once we understand the molecular weight, so then the right standard can be used for quantification. And another challenge is once we understand the molecular weight. For example, if you see here, several grades are having same molecular weights, and hence there is always an ambiguity like which grade to be used for quantification. And if you see E4M, K4M, and F4M all are having very close molecular weight, and its substitution changes based on methoxy content and propoxy content, and hence the degree of substitution understanding is very much essential in these products. So although we understand the molecular weight and its distribution profile by size exclusion chromatography coupled with moles, and as a concentration detector, RA can be used to get the right peak profile where there are no standards required for quantification. And as several grades are present with similar composition, so the degree of substitution is very important to understand. So for to understand the degree of substitution, so which where we have used HSQC NMR as a tool for hypermellose differentiation. When we ran the HSQC NMR, with we can able to see O methyl signals and the methyl signals from hydroxypropyl cellulose group, where the arbitrary number can be calculated from these ratios, and then each grade gives a very unique number. Based on that, the quantification can be determined, and the type of the polymer can be understood. And so overall, to summarize, it is very important to understand the right grade of the polymeric standard with its right substitutions using the size exclusion chromatography coupled with light scattering detector, and also as NMR. So, as another example, as a case study, which I'll be presenting on polysorbate 80. Polysorbate 80 structurally, it undergoes degradation upon hydrolysis and autooxidation. It gives into a different fatty acid chains and peroxides, aldehydes, ketones, etc. So where it is very important to understand to use the right tool for quantification of polysorbate 80, and also it is important to understand the extent of degradation to get compensated for the accurate quantification of polysorbate 80. And this is the LCMS ESI profile of different polysorbate standards which are used, and also from a reference sample for quantification, and where we see the different degradants of polysorbate 80. So upon understanding of all these, a quantification can be understood, and can be easily estimated. The main challenge here is it undergoes degradation with time, hence this is one of the major challenge. And also polysorbate comes with different grades, which is basically super refined, and the high purity, which is basically changes um, in terms of its slightly fatty acid compositions. Where to understand the grades, one of the important tool which can be used is the gas chromatography coupled with FID detector. And the based on the peak profiles, it can be easily understood upon base hydrolysis followed by derivatization with to have the methylation to get the volatility of these fatty acids, and then separating into a gas chromatography using FI detector. These are the experimental conditions which we have used to separate all the peaks, and based on the peak ratios, it can be easily understood which of the grade is used in the reference product. So overall, these are some of the techniques which I have just tabulated to understand and quantification purpose of different excipients using different analytical techniques based on the availability from the laboratory. It can be easily used, and then to get an accurate information of the excipient. So, the overall, the current challenges to summarize and also to understand the future directions, the selectivity and specificity of the analytical technique is very, very important. Where it is important to use at least um, the negative experiments to prove that the method is selective, the technique is selective, and have very specific to the analyte of interest. And also some of the excipients has under its absorption behavior is important to understand for accurate quantification, and the degradation behavior is also very important to understand to get compensated to get the actual original composition of the product. So based on our experience, the SEC coupled with multi-angle laser light scattering, where along with RI, either UV or viscosity detector is the best choice for characterization of structural attributes of all the polymeric material. And it is important to understand to have a suitable standard for quantification in case of polymeric materials. Otherwise, the response under the peak may change. And also ensuring the availability of structurally similar standards in case of conventional SEC techniques are used, as we going to see variability across the lots with time because of the degradation, absorption behaviors, etc. So analysis of multiple lots of RLD with different shelf life is important. And so that is where we can correlate the data with the age of the sample and then understand better. And use of orthogonal techniques is very, very important. At least couple of techniques can be explored to understand and cross verify the data based on structural aspects of each of the molecule. And another important aspect, but not the least, is the development of right skill set on all these advanced analytical techniques, which can help to get right at first time. Thank you. So if you have any questions, I would be very happy to take it up.